Hello, I'm Paul Krogstad from the UCLA Departments of Pediatrics and Molecular Pharmacology. I'm happy to have this opportunity to tell you about our exciting research and to enlist your support for our efforts to develop broad-spectrum antiviral medications to treat enterovirus infections. The enteroviruses are a group of more than 100 viruses known to cause diseases such as poliomyelitis and other infections of the central nervous system. It's nearly certain that everyone watching this has some experience with them, as they cause 10 to 15 million cases of infections per year, including the majority of summertime colds, brief episodes of gastroenteritis, and flu-like illnesses. This occurs because enteroviruses are readily transmitted from person to person. All people in the United States can have these sorts of infections, but infants and young children are particularly vulnerable to the most serious complications. And in fact, up to a 40% mortality risk occurs when newborn infants acquire them. Despite the frequency of these and the fact that they infect children and adults, it is surprising that we have no approved treatments. Treatment is purely supportive at this point. In response to this challenge that occurs perennially, we have discovered a small molecule with broad spectrum activity against enteroviruses and are looking to try and develop this into a potent therapeutic agent. Now, as I mentioned, although brain infections are usually the most severe, the actual types of infections that you see vary a bit internationally. In the United States, meningitis occurs in about 75,000 individuals per year in the United States, according to CDC estimates. But in contrast, in Southeast Asia, one of the most prominent concerns about circulating enteroviruses is a variation of the common childhood disease known as hand, foot, and mouth disease, in which the infections damage a part of the brain stem in children and young adults and can produce a variety of ailments that include acute paralysis, a loss of control over blood pressure, and in fact, a loss of control over breathing as well. And as I mentioned, newborn infants throughout the world are particularly susceptible to infections that resemble bacterial sepsis. And it is one of these that in fact led me in recognizing just how frequent these are in the world to move into this area of research. The case that I'd like to mention that drew me into enteroviral therapeutic research is that of an infant who was born at UCLA Medical Center, but developed fever and irritability on the third day of life. Cardiac arrhythmia developed on the fifth day of life, and he was moved to an intensive care unit where an X-ray demonstrated that his heart was enlarged and dysfunctional, and we confirmed an enterovirus infection by viral culture. Unfortunately, other organs began to be affected. His liver and his liver failure and kidney failure soon developed, and although he received the maximum level of ICU support that was possible, he died at 4 weeks of age from heart failure. It's dismaying that although we now have dozens of antiviral agents available to treat other serious viral infections, none of these agents have any activity against enteroviruses. Faced with this, we began to seek an answer to this perennial challenge. Who are we? Well, I've mentioned that I am a specialist in pediatric infectious diseases. I have 30 years experience as a virologist and clinical investigator as well. My partner, Mike Jung, is a renowned synthetic chemist whose experience in medicinal chemistry includes the development of anti-infective agents and of drugs to treat cancer, including Xtandi, used throughout the world for the treatment of prostate cancer. What we have done instead is identified a small antiviral activity against a highly conserved enteroviral protein known as the 2C protein. The key advantages of this is that it's very well conserved among the more than 100 enteroviruses. And in our testing, we see that inhibiting the activity of this protein prevents the growth of more than 50% of the enteroviruses seen in the United States. In our preliminary testing, it appears to have very low toxicity, and viruses are unlikely to develop resistance during brief exposures to this, we think, because because of how rapidly suppression of the activity of this one enzymatic protein in enteroviruses leads to just a complete cessation of virus replication. If we were successful, this would be the only drug active against a broad spectrum of enteroviruses. Nothing else that has been in development or has been described has this kind of spectrum. It also potentially has the ability to treat other sorts of viruses because proteins that are structurally similar to the 2C protein are seen in other human pathogenic viruses. The way that we reached our current state of development is pretty straightforward. We started with a relatively small library of compounds, including common pharmaceutical compounds, and worked using a screening assay that involved a virus very similar to the one that I described in the case of this unfortunate newborn at UCLA. Following that initial assay, we had a second antiviral assay in which a fluorescent version of the virus was used to screen out some of the background, and two additional types of assays that looked at the amount of virus that was actually made in the presence or absence of all of these compounds. We ultimately realized that the screen brought out 21 Pyrazolo pyridine carboxamides, PPCs as we call them. Working with Mike Jung, we did a structure and activity relationship analysis on these compounds, and he went on to synthesize nearly 60 additional variants of this. We did some initial limited solubility testing to be able to develop a soluble formulation, and we have gone as far as some initial pharmacokinetic analysis in vivo. To give you an idea of the potency of these antiviral agents, I show you the structure of the first compound, JX001, and as well as its activity against a wide array of 15 different enteroviruses with a total of 100. These represent some of the most common viruses circulating in the United States. Now, on the leftmost column, you'll see that I have fluoxetine demonstrated, which is Prozac, and the color scoring system shows you low activity as red and high as a deep blue. Neither fluoxetine or the antiparasitic agent mefloquine have consistent activity against poliomyelitis at all. That's PD1 and 3. And mefloquine is particularly anemic when it comes to acting upon some of our most common US enteroviruses. But the four compounds on the right all had fairly broad activity against this panel of 15. Only one of them had much effect on poliomyelitis, and that's the compound that we're showing on the right, which is JX001. So beginning with this compound, we went forward to develop others, and all based on the pharmacophore, the central structure in JX001. One example of this is JX017, which has better toxicity and antiviral activity in vitro, and we have taken this as far as doing limited pharmacokinetic testing in mice. As you see in the right panel, enteral exposure, meaning gavaging the doses into the groups of mice, we see produced peak concentrations that were near the target that we would expect based on our in vitro testing that is needed to have antiviral activity. Our current state of activity, our current state of research is to further improve upon the solubility formulations so that we can go back and test a variety of doses and eventually do virus challenge experiments in well worked out murine models that have been used to examine infections of the brain and heart in mouse models. One other important consideration that would allow us to move forward is that intellectual property rights pertaining to the JX compound series have been established. So I look forward to your questions and inquiries that you might have about the further development of the JX compounds, and I look forward to hearing from you.